Inside every human cell, irrespective of tissue type, important biological functions are mediated by cytoskeletal proteins. The ability of all human cells to carry out complex functions such as cell migration, intracellular organelle transport, mitosis, and cell division relies on the cytoskeleton. Cytokinetics, a cell biology-driven drug discovery company, is dedicated to the discovery and development of novel, next-generation therapeutics that target newly characterized cytoskeletal proteins. The cytoskeleton is a dynamic architectural framework in human cells and is comprised of a network of protein filaments and other related proteins. Actin filaments are used for short-distance intracellular transport and are essential for cell movement and muscle cell contraction. Microtubules from another cytoskeleton filament network is used for longer distance transport. Microtubules radiate from the centrosome at the center of a cell and consists of a linear array of protein tubulin. Both actin filaments and microtubules serve as a system of tracks within the cell upon which motor proteins move, thereby transporting a variety of cellular cargo. These motor proteins, filament networks, and other associated proteins all function together and form the cytoskeleton. The cytoskeleton is one of a few broad biological areas with the potential to yield novel pharmaceuticals across a range of human diseases, including cancer, a disease of unregulated cell proliferation. Inhibiting cell division is a recognized strategy for treating patients with cancer. At the core of cell division is the mitotic spindle, a structure formed largely of microtubules. The spindle mediates the ordered series of mechanical events responsible for delivering identical copies of the genome to each of two daughter cells arising from cell division. Proper formation and function of the mitotic spindle is required for cell cycle progression through mitosis. The mitotic spindle is an important target for modern cancer therapy. Several existing cancer therapies inhibit dividing tumor cells by targeting tubulin, thereby perturbing microtubule dynamics. However, tubulin and microtubules are present in all cells, including neurons, and play an essential role in many cellular functions beyond mitosis. Unlike tubulin and microtubules, mitotic kinesin motor proteins play an essential and exclusive role in cell division and are only expressed in proliferating cells. These proteins appear to play no role outside mitosis. Kinesin spindle protein, also known as KSP or EG5, is a mitotic kinesin which acts at the earliest stage of spindle formation. Early in mitosis during prophase, KSP forces the emerging spindle poles to move apart, driving formation of a bipolar spindle and enabling chromosome segregation into two resultant daughter cells. KSP is not expressed in neurons and has only one known function: to drive spindle pole separation during mitosis. Inhibition of KSP motor function prevents formation of a bipolar spindle. KSP inhibition results in cell cycle arrest and mitosis with a characteristic monopolar spindle on which chromosomes are arrayed. In cancer cells, duplicated chromosomes remain attached to the monopolar spindle in a persistent state of cell cycle arrest, resulting in programmed cell death or apoptosis. Inhibitors of KSP and other mitotic kinesins may therefore represent next-generation antimitotics by specifically targeting the mitotic process and inducing cancer cell death. Cytokinetics and GlaxoSmithKline are engaged in a strategic alliance to discover, develop, and commercialize small molecule therapeutics that target human mitotic kinesins for applications in the treatment of cancer and other diseases.